That's okay. That's what we were talking about. So we covered type 1 and type 2, type 1 and type 2 hypersensitivity, right? And so we gave example of type 1 hypersensitivity being allergy, type 2 hypersensitivity being the transfusion reaction, and now it's time for type 3, which is kind of we can say a true immunopathology, because if you think about that, transfusion reaction is largely iatrogenic. Allergy is a pathological reaction, but we don't consider it being like wildly horrible, right? I mean, who has allergies? I have allergy. Okay, you know, people have allergies. People live with allergies. Type 3 hypersensitivity, on the other hand, is about pretty serious disease of epithelial tissues, right? We didn't talk about it, did we? I don't think so. We didn't. Okay. So here how it works. Let's first recognize what the epithelium is. So epithelium, where can you find epithelium? And what is the function? Okay, it's lining that separates what and what? A what? Well, for instance, where are you going to find epithelial covering? Okay, respiratory tract, throat, digestive tract, blood vessels, kidney tubules. What's common in terms of between digestive tract, respiratory tract, blood vessels? What are they? What are they, shape-wise? Huh? They tubes, right? They have lumen in the center. And epithelium separates the lumen and the tissue. Does that make sense? So that's just to give you an idea. So that's the point. That's the function of epithelium, to separate. So and the epithelial cells, they are attached to the basement membrane. And then you have a tissue here. Okay, and those are epithelial cells. Does that make sense? Okay, remember that? So, in type 3 hypersensitivity, antibodies bind to the proteins found in the basement membrane. Collagen, laminin, and so on and so forth. And remember, antibodies, do they lyse anything? Do they destroy anything, antibodies? They do not. But they tag, right? They aim other components of immune system to the pathological, to the site of injury. So essentially these immune complexes target macrophages and neutrophils to destroy the basement membrane. Does that make sense? So neutrophils and macrophages, basement membrane destruction. Does that make sense? Now, if you destroy basement membrane, what's going to happen to the epithelium? It's not even blood cells will start just kind of fall off, separate. There will be the integrity of the epithelial tissue will be disrupted. Okay. Now, what does that mean? For instance, in the blood vessels, what does that mean? What will happen? Plasma leakage, right? And inflammation of the blood vessel walls will lead to thrombosis. So leakage thrombosis. Kidneys, glomerulonephritis. Joints, inflammation of synovial membrane will lead to the destruction of synovial membrane, first of all, at the same time, accumulation of the fluid in the joints. So joints will become inflamed, and that's arthritis. Does that make sense? Now, it's not the only symptoms, but those are kind of notable symptoms, very common symptoms of the disease that you may know as lupus, okay, or systemic lupus erythematosus, okay. So that's the typical example of type 3 hypersensitivity, when immune complexes are formed in the basement membranes of epithelia all around the body. Okay? Does that make sense? Is that clear? In type 4 hypersensitivity is also called delayed, because it is mediated by T cells. And as you can imagine, to develop the T cell response, it takes some time. That's why it is called delayed. Does that make sense? Now, I usually provide, so we'll do TB test, because it kind of things that you're going to deal with when you are going to be in the healthcare. But there's another very good example of type 4 hypersensitivity, is the reaction to poisonous plants like poison ivy. So how TB test works? First of all, what does TB test detect? It detects the tuberculosis, the presence of immune response to tuberculosis. So either previous exposure to tuberculosis and acquisition of the microbe, or current infection. Okay, and there are some considerations, okay, to it. So what happens when you are first exposed to tuberculosis? Okay, that's Mycobacterium tuberculosis. Okay, we'll call it MTB, Mycobacterium tuberculosis. Your immune system, your dendritic cells, okay, will acquire Mycobacterium and present it to the T cells. Does that make sense? And your body is going to form the population of T cells. So it's going to be TB specific T cells. Okay? Does that make sense? Does that make sense? You're going to form a memory population, right? Even though you may not have an active infection. So what is done? How is the TB test done? So you get an injection. Nah, not subcutaneous. It's intracutaneous, intradermal, bless you. It's intradermal. So injection with mycobacteria doesn't go under the skin. It goes inside the skin. Now the reason for this is you have special cells in the skin that are called Langerhans cells. They are the dendritic cells in essence. Okay? And when you get the injection with the tuberculin, Langerhans cells will present the tuberculin on its surface. Now, is there a similarity between the green dendritic cell right here and the Langerhans cell right here? What's the similarity? They present the same antigen, okay? They present the same antigen, which means that now this Langerhans cell, by presenting the tuberculin antigen, will stimulate the proliferation of memory cells, memory T cells, that are TB specific, okay? Now, if you remember T cells, what can they do? There are two types of T cells, which are helper and killer. Okay, cytotoxic, yes. So you're gonna have CTLs and you're gonna have helpers. So CTLs will cause apoptosis in the site of injection. Helpers attract macrophages, okay? And it's gonna increase inflammation at the injection site. Does that make sense? Now, it all contributes to the formation of the bump at the injection site. Does that make sense? What I was about to say. The more severe your response is, the bigger the bump, the more robust T cell response you produce, which means if it's really large, it's likely that you have an active infection. If it's smaller, you may have been exposed. There are clinical criteria in terms of the height of the bump and diameter of the bump that will allow the doctor to differentiate between the, having and not having the TB. So one of the problems with this test is that it detects if your immune system was ever exposed to the tuberculosis antigen. Does that make sense? Is there any circumstance in which the person can be exposed to the tuberculosis antigen without actually being infected? Yes. There is a vaccine, which is not used in the United States. It is called BCG. I feel absolutely horrible. I look up regularly what this acronym stands for, and I know that B stands for bacillus. CG are the last names of two French guys. I just cannot memorize those last names, okay? But it's BCG vaccine, which is used in many countries, including my home country, including Russia. It is used in India, in Africa, in England, virtually everywhere in the world, but not in the US. The reason for use, it decreases the chances of kids to acquire TB. Okay, it's not really extremely effective, but it nevertheless works. And if you can spare kids from acquiring tuberculosis, it's worth doing that. Okay, and it is done at the age of like 3 or 7 days. So usually kids do not have a lot of decision-making power at this age. In the United States, the reason why it is not done because it can mask the actual the results of TB test, skew it, showing that okay, this guy has a TB. Well, this guy doesn't have a TB. This guy was exposed to the antigen by vaccination. Okay, so that creates real problem for people from India, people from Russia, people from Europe when they have to go through the TB tests in the clinic for some reason, okay? And it's every time it's like a Russian roulette. Are you going to come out positive or not? Because it's not 100% effective. Does that make sense? So sometimes you can come out negative, although you have been vaccinated. Does that make sense? So in the brief, same goes for poison ivy. Your immune system gets exposed to the antigen by contact, and then upon the second exposure, after a while, you develop that massive inflammatory response in the skin where poison ivy is present, where the toxin is present. Does that make sense? It bubbles, yes. Langerhans cell, it's a type of dendritic cell. Does that make sense? It's pretty much found in the stratum spinosum of the epidermis. So things like mosquito bite, poison ivy, everything that gets on the skin is being presented. Any antigen that gets on the skin is being presented by Langerhans cells. Does that make sense? Okay. I try to remember because my computer, it took it something like 25 minutes to reboot, so I'm not really optimistic about its capacities. I think our next topic of discussion is transplants. Okay. You should check, if you have like a study guide, check me in this. So transplants. There are 4 different types of transplants, and generally in medicine, the transplant is has a word graft, right? So it's a grafts. One type, and that's kind of common, is autograft. When the tissue is transplanted from yourself. Can you give me an example of autograft? When people receive autografts, when people receive their own tissues, skin transplants, huh? Skin grafts, yes, for like wounds, burn victims, yes. A lot of it, I had a privilege to listen to a wonderful lecture on head and neck cancer. It wasn't so much focused on head and neck cancer, it was mostly focused on the problems associated with the skin graft, with the transplantation. But it was fascinating to see how disfiguring the cancer can be by itself, first of all. Second, what happens, what people, for example, people's face, people's jaw look like after the surgery. People may lose most of the upper jaw, lower jaw, different parts of the head, okay? And then what cosmetic surgery can do in terms of the restoration, right? And of course they do the skin grafts. Okay, so that's autograft. Another one is isograft. So if you have identical twin, maybe it's a good time now to lock the twin in the basement and use him or her as an organ donor. Okay. Only if you have identical twin, because that means that you two are pretty much genetically the same. You have same genes for major histocompatibility complex, which actually is responsible for rejection of the graft. Does that make sense? Now, this is not a very common situation, and if you are a heavy drinker, then even though your identical twin may provide you a piece of his or her liver, eventually your identical twin will run out of liver or other organs. Okay? So it's not a sustainable source of organs. Now, another type of the graft is allograft. When you have somebody that is not an identical twin. Okay? Which means, does it mean your brother or your sister or your mother? Yes. It's not identical twin, it's still allograft. Oh yeah. What I mean is, when there is an allograft, in some cases, somebody completely unrelated to you may turn out to be more antigenically similar than your immediate family member. Does that make sense? And finally, xenograft. So that's the transplantation from, and the only animal that I can actually draw is the mouse. Of course, nobody does a transplantation from mice, but it's transplantation from animal to a human. Maybe pretty common are heart valves. From pigs, yes. Pigs are probably the most immunologically similar to humans. And they great models because they have similar size of virtually everything. And I spoke with one grad student from University of Indiana who studied the cardiovascular disease in pigs. I mean, it's not really different from what we do in mice, or well, nobody does in monkeys, but the terms of work are really fascinating because she mentioned it's kind of hard to properly anesthetize the pig, about 50-60 pound piglets. Okay, so you have to really watch if the piglet is waking up, because if it does wake up, then it's going to jump off the cart and just run down the hallway. In this case, yeah, in this case, we just had a lunchtime conversation, and she said, in this case, you just scream down the hallway, shut the doors, because if it runs into the lab, it just ruins everything. It doesn't attack people, it just panics, and then you have to chase it and catch it and bring it down pretty much. So those are 4 types of grafts, and you have to know pretty much the description of each. I will ask you, transplantation of from here to here is this type, okay, from animal to human, blah, blah, blah. Now, major problems with grafts. There are 2 problems. What is the main problem with the grafts of solid organs, like liver, kidney grafts? Rejection, yes. So, in rejection, the problem is that your immune system, right? For example, B cells or T cells, NK cells, T cells, they will not recognize the MHC. Does that make sense? Major histocompatibility complex. I mean, they will recognize it as foreign. All right? So you have a cell with a receptor, and then you have some sort of a tissue, and you have MHC molecule here. And there's no recognition, immune rejection. Does that make sense? So essentially, graft will be not quite physically destroyed. There will be a massive inflammation. Massive. Okay? And in order to prevent this response, we use lots of drugs that do what? Just generally. Huh? Suppress the immune system, right? Does that make sense? So people who are after the transplant, they severely immunosuppressed, and they're actually at risk of high risk of infections. Yeah, and I don't remember if I mentioned the problem with the rabies in the transplant, did I? No? There was a famous case when the person died in a car crash or something, and his organs were used for transplantation. He was an organ donor. The problem is that he was in incubation period. He was infected with rabies. And rabies can be hidden for days and weeks and months, actually. So he had rabies virus in virtually every organ of his body, and these organs were transplanted to other people, and you can guess that every recipient died because of rabies. It was a pretty horrific story. Huh? Yeah, but the problem is we don't know what to test for. You know, here's the point. If you're in transplantation, I don't want to say business, but let's say transplantation business, and you have organs fresh from the person who just died in a car accident, what are the chances that this person is going to have rabies without any clinical symptoms or anything? They're really low, actually. So they never test. Yes. Natural killer cells. Okay. So yes. For rabies, as far as I know now, they do. But we respond reactively, not proactively. However, there are several diseases that donors are routinely tested for. One of them is cytomegalovirus. Just as an example, so CMV can be latent, okay, in epithelial cells or monocytes. Here what happens, say you have a donor, kidney donor, okay? You have a recipient, and kidney donor has latent cytomegalovirus. There are no symptoms whatsoever, absolutely, period, zero, okay? So this kidney is getting transplanted to the recipient. Good? Kidney is being transplanted to the recipient, and the recipient starts to receive immunosuppressive therapy. Since there is no immune system is weakened, cytomegalovirus starts to replicate. It doesn't destroy, like, you know, kidney, the transplanted kidney doesn't melt down. It causes inflammation. And it causes inflammation, what is fascinating, in the artery, in the renal artery. So renal artery starts to stenose due to the inflammation. Blood flow to the kidney decreases. Transplanted kidney becomes hypoxic. And boom, that's it. It doesn't work anymore. Cytomegalovirus infection is the leading cause of kidney rejections. So they test every kidney donor for CMV and another Epstein-Barr virus. Does that make sense? Okay. Now that's for solid organs. What about bone marrow transplantation? What about treatment for leukemia? Okay. So bone marrow. When is a bone marrow transplantation happening? And do you know how it goes? I mean, how people are getting treated for leukemia? You know, what do they start with before transplanting the bone marrow? Well, not everything, just bone marrow. Because when you transplant, like when somebody has a problem with the liver and you want to put in the new one, you don't put it like on top of the old one. You want to remove the old one first. Same goes for bone marrow. You want to remove the old one, the diseased one first. With the drugs, radiation, so you pretty much totally destroy bone marrow. Got it? What is the function of bone marrow? What does it do? What does it produce? So red blood cells, platelets. So it produces what? One word. Well, two words. Platelets, red blood cells. What else? Huh? Blood cells. Very good. Blood cells. All blood cells. Red blood cells, platelets, white blood cells. Does that make sense? Bone marrow is gone. Do you have any immunity? No. It's all gone, right? So they put new bone marrow in you. Okay? And does it now produce red blood cells? Well, let's say the transplantation went well. It does. What about white blood cells? Sure enough. So now imagine you're a white blood cell that was transplanted in the new body. You come out, what do you see? A ton of foreign tissue. Does that make sense? So you've got T cells from transplant that recognize the MHC from recipient. Not, well, they recognize it as a foreign, right? So this all creates what type of situation? What these white blood cells produced by the transplanted bone marrow start to do. So it's not, well, it's kind of autoimmune. You develop your immune system, new immune system, transplanted immune system, starts to attack your own body. Does that make sense? The new incoming white blood cells, they start to attack everything, every part of your body. And this is called graft versus host disease. GVHD. It's essentially, it's a multiple organ failure. Really, really bad disease. Does that make sense? Okay. So what you have to know for rejection in GVHD, sort of the mechanism. So what happens where? In rejection, the immune system of the recipient attacks the solid organ that's been transplanted. In graft versus host disease, immune system that comes out of the transplanted bone marrow starts to attack I just got a brain freeze. The tissues of the recipient. Okay? Immunosuppressants. So people who receive the bone marrow transplant, they are on the permanent immunosuppression. My wife's colleague, he was treated with leukemia. This is about like 25, 26, I think now. I mean, he's a healthy man, okay, but he's on immunosuppressive drugs all the time, and he doesn't eat at the cafeteria. And I must say, it's a decent cafeteria. And if somebody sneezes in the lab, he just collects his belongings and goes home. And everybody understands why, because he's not supposed to be exposed to any kinds of bugs. He doesn't want to test if that's just a simple common cold, or it's a flu that can put him in hospital. So they do, they study Crohn's disease, inflammatory bowel disease in mice. So they work with a lot of germ-free animals, animals that have sterile intestines. It's pretty big lab, so like if somebody sneezes on one side, you may not even hear on the other, but still, if he realizes that somebody can expose him, And that's fine, because you can't stay home every time you have sneeze because one of your coworkers is immunosuppressed. It's just not possible. Okay, but that's something, to think about. Different lifestyle. Does that make sense? Well, last thing to talk about is immunodeficiencies. So we pretty much, so far, look at this. We talked about hypersensitivities, immune system being hyperactive. Now we're going to talk about immune system being not active. Okay. That's a mouthful. There are two types. It can be acquired and congenital. So acquired, what does that mean? Yes, but more inclusive. What? You aren't born with it. It happens later. Yeah, it happens later. Okay. What examples of acquired immunodeficiency you can think of? Yes, HIV/AIDS. Now, the kind of word of warning, remember that HIV is the name of the virus. AIDS is the name of the disease that this virus causes, or may cause, if not treated. T cell, CD4 cells, a gun, that's AIDS. Okay. That makes sense. Any other examples? Something else that depresses the immune system. Huh? Cancer. Yep. Cancer. If solid organ cancer will depress the immune system. Leukemia is, although there are a lot of white blood cells, they're all dysfunctional pretty much. Right? Any other? Well, there's one disease that causes you to be immunosuppressed for about 6 months. It's called measles. Okay, it kills all the B cells. Just wipes them off. So you don't have antibody response for about 6 months after you recover from measles. Now, what about congenital? Now, these guys, you are born with them, right? Now, what two major branches of immune response do we know? Okay. You are talking about two branches of adaptive. So we have humoral and we have cellular. So this is going to be, that's going to be adaptive, right? Now, you mentioned innate. In terms of the deficiencies in the innate immune response, they are usually subtle. Okay? So we're going to put it subtle deficiencies. What do I mean by subtle? There was a case of a girl, I might have mentioned it, I don't really remember. A girl was admitted to a hospital at the age of like 6 months with extremely severe influenza. The girl, otherwise, nothing pointed that she would have such a severe influenza, even though she was 6 months old. So she had just unusually severe disease. It was a few years ago, so doctors, along with the treatment, so they actually saved her in the end. but they also wanted to see why the disease was so severe, and they sequenced her genome, which at that time was something like 10,000 bucks. I mean, it's not cheap, but it's feasible. And it turns out that she had, I think, single mutation in one of the genes that is induced by interferon. So she had very subtle abnormality in the interferon response to the infection. And then they showed that that particular abnormality in animals contributes to abnormal pathogenicity and blah blah blah. Does that make sense? So she recovered, she completely recovered, but now her parents know that she has a certain immune deficiency that makes her more sensitive to influenza virus infection. Does that make sense? So those, because if you think about this, if you have a mouse that, well, mouse can lack interferon response, but if you have some animal without complement, this animal is probably going to be embryonically dead, so will not be even born, okay? Because those systems are very broad spectrum, okay? Knocking them out will affect very, very many aspects of survival. Now, in terms of humoral and cellular, turns out people can lack either, okay? So when they don't have humoral response, what do they not produce? When they do not have humoral immune response, which molecules they cannot produce? Which produce what? Well, which become plasma cells that produce which molecules? Huh? No. Well, they probably do, but it's not their main function. Plasma cells, huh? Antibodies, yes. So, no antibodies. This is called a gamma globulinemia. That's a long word, which means no gamma globulins in the blood. Can you read the chicken scratch? A gamma globulinemia. Emia stands for blood, A stands for no, gamma globulin is antibody. Does that make sense? There are different degrees of a gamma globulinemia. Sometimes people just don't produce B cells at all. Sometimes B cells are produced, but they're not functional. Sometimes there's a problem in antibody synthesis. Right? Cellular response. No T cells, right? Again, there are various types of severity. The one that I want to mention to you is called D. George syndrome. It's a lack of T cells. Does that make sense? Does that make sense? We good? Can people have, is there a chance that people have no B and T cells? Yes. So this condition, when there's both are absent, is called severe combined immunodeficiency, or SCID. Now, if you've heard about boy in a bubble, that is the case of SCID, severe combined immunodeficiency. I am aware of two cases that I learned about and I read a little bit about. For some reason, I think they both were handled in Texas, although I'm not sure about geographical location. So those kids were essentially raised in the hospital in a special chamber. The positive pressure, now it's not like chamber like a room chamber. They have something like curtains that surround them, and they have a positive pressure flow. Okay, so air goes away from them to keep the potential bugs away. People that attended them had to wear special outfits and everything. Does that make sense? The food was sterilized, special food. I mean, it's pretty expensive, right? Now, when they reached certain age, they were treated. The guy number 1 was treated with the blood transfusions, which provided him at least the B cells, okay? At least some immunoglobulins. And he actually turned out to stop living in the hospital, okay? He still was severely immunocompromised, but blood transfusions allowed him to live more conventional life. And he died in his 30s because of the kidney failure. With every blood transfusion, there's a certain percent of hemolysis, which releases iron and heme in the blood. And that slowly but steadily kills the kidneys. Does that make sense? The story of another boy, a little setter, well, not a little actually. So when he reached certain age, I don't remember what age was that, something maybe like 12, 13, they decided to do the bone marrow transplant, which was, I think, a brilliant idea. Because you do the bone marrow transplant, the boy starts to produce all the normal cells, okay? He's protected against infections. He has to live on immunosuppressants, so it's a fine line, you know, it's a balance, but he can be like any other patient who receives the bone marrow transplant. So the boy receives the transplant from his sister, who turned out to be very genetically similar to him. One thing that his sister wasn't tested for, the presence of any latent herpes viruses. And she turned out to be Epstein-Barr virus positive. Epstein-Barr virus resides in B cells and in bone marrow cells. So when bone marrow was transplanted, and Epstein-Barr virus, which was latent, started to kind of, well, it's from the standpoint of the virus, this guy has no immune system. I'm having a field day. So virus started to replicate uncontrollably, and at that time there was no good drug to prevent the infection. Now we have it. It's not awfully good, but it's very decent. So essentially, shortly after transplantation, the boy died of the systemic Epstein-Barr virus infection. His parents decided to have another child, and the second boy died at the age of 6 months because of the same condition. And then they decided to have a third child, although, well, there was third attempt to have a boy, although doctors suggested not to, but they persevered, and the third boy turned out to be healthy. That's a great question, because what I was about to ask you, this condition, I mentioned, you see, there were no girls involved in the disease. So my question is, what type of inheritance? Huh? What? Sex-linked? And, huh? Sex-linked. Dominant or recessive? Recessive. Sex-linked recessive. Now what it means, that's the mother, right? And XY is the father. So first two boys, first two boys had this genotype. Does that make sense? Right? And the third boy simply had this genotype. He just didn't receive the chromosome with the pathological gene from his mother. And that's why he was fine. So essentially, this condition, women are carriers and men are sick. You can think of a situation when you can produce a sick woman, but it's really hard to pull off. Because there should be a mating between the carrier female and the sick male. And it may not work from the first try, so there should be several tries. I mean, it's not really feasible. Does that make sense? Terrific.